Afternoon one and all. So, yes, in yesterday's class we have discussed about non-membrane bound organelles and the two organelles of endomembrane system. So, the next non-membrane bound organelle which is found only in the animal cell, this non-membrane bound organelle is found only in the animal cell, that is centriole. Okay. Centriole is found only in the animals. So, here if you see centriole is present inside the centrosome. Centriole is present inside the centrosome. Centrosome is surrounded by pericentriolar space. Fine. Inside the animal cell cytoplasm, you have centrosome which is surrounded by pericentriolar space, and inside the centrosome you have two perpendicular structures called as centrioles. These two are perpendicular to each other. Fine. If you have to see the microtubular arrangement of this centriole, the microtubule arrangement will be centriole microtubule arrangement will be n plus 0. Okay. First we will look into the arrangement of microtubules inside the centriole. What will be the microtubular arrangement? At the center if you see there is a proteinaceous hub. Okay, there is a proteinaceous hub at the center. To this proteinaceous hub there are 9 radial spokes which are radiating out of this proteinaceous hub. To each radial spoke there are 3 peripheral microtubules. Okay. There are 3 peripheral microtubules adjoined to each radial spoke. Okay. So, these 3 peripheral microtubules can be termed as A, B, C. Microtubule of one radial spoke is connected C microtubule of other radial spoke. These are AC connections. Okay. AC connections, these are called as AC connections, and these are connected by the protein nexin. Fine, these are connected by the protein nexin. So, at the center there are no microtubule, at the center it is proteinaceous hub, there is no microtubule. From here 9 radial spokes are radiating out and each radial spoke is having 3 peripheral microtubules. Okay. So, and each microtubule of this peripheral radial spokes are having what connections ma? AC connected by nexin protein. Now, if you have to see what are the total number of microtubules present in the centriole. In centriole if you see, that means in a single centriole if you transversect it and observe, you will see that microtubular arrangement. At the center anyhow there are no microtubules. To 9 radial spokes, to each radial spoke how many peripheral microtubules are there? 3 peripheral microtubules. So, for one centriole how many microtubules will be there ma? 27 microtubules. Okay. 27 microtubules in one centriole. Fine. If the question is in one centrosome, do not get confused between the terms centrosome and centromere. Centromere is the primary constriction that you see inside a chromosome. In a chromosome, the primary constriction which you see, this primary constriction is centromere. Centrosome is an organelle which is a non-membrane bound organelle, holds out 2 perpendicularly placed centrioles. Centrioles are cylindrical structures which are non-membrane bound, which are present only in the animal cells. Okay. So, in a single centrosome if you see, how many microtubules will be present? 2 centrioles are there na in single centriole? Of course, it is 27 plus 27, it is 54 microtubules. Okay. So, this is the microtubular arrangement. What will be the function of a centriole? What is the function of a centriole? Centriole function is formation of spindle apparatus. What is centriole function? Formation of spindle apparatus and helps in cell division. Okay. Centriole function is formation of, take down parallelism, formation of spindle apparatus. Fine, right? So, this is about centriole and centrosome. fine. And next if you see flagella and cilia. Flagella and cilia also has microtubular arrangement. Look into it. Flagella and cilia. They will be having 9+2 microtubular arrangement. Okay, what is this 9+2 microtubular arrangement? Look at here. At the center you have two central microtubules. Okay, two central microtubules at the center. They are connected by a proteinaceous filament again. These two central microtubules are surrounded by a central sheath. Okay, two central microtubules are surrounded by a central sheath. Adjoining to this central sheath, we have 9 radial spokes. Okay, 9 radial spokes to this central sheath. Each radial spoke will have 2 peripheral microtubules. What are they? A and B you can say. B is connected to A. So, here you can see in a cilia or flagella we have AB connections. Fine, we have AB connections. Okay. So, at the center you have two central microtubules surrounded by a central sheath. Central sheath will have 9 radial spokes. Each radial spoke will have 2 microtubules. Clear, right? So, here you will see how many number of microtubules. 2 central microtubules, 2 plus number of microtubules. 2 central microtubules plus how many radial spokes? 9. Each radial spoke is having 2 microtubules. So, total it is 2 plus 18, 20 microtubules. 20 microtubules you observe in what? Cilia and flagella. And here if you see in a flagella, if this is a bacteria, flagella originates from the cell membrane and they are the extensions of what? Cell wall. They are the extensions of cell wall. In the cell membrane and cell wall region, this region is called as what? Basal body. Basal body. And this is called as hook. And this is long filament. Okay, we have basal body, hook and filament, right ma? So, the basal body is connected to the body of the prokaryotic cell by what arrangement? 9+0 arrangement, because this is connected by the help of a centriole. Okay, centriole will connect the basal body of the flagella to the cell. So, the basal body microtubular arrangement will be 9+0. Fine. Hook and filament will have 9+2 arrangement. Fine. Remember this. And prokaryotic flagella is made up of flagellin protein. It is having 9+0 arrangement of microtubules. Eukaryotic flagella, which is observed in some protozoans, like if you see flagellate protozoans, for example trypanosoma, euglena, okay, these are eukaryotic unicellular bodies, right? They will be having 9+2 microtubular arrangement. So, here if you see flagella, prokaryotic and eukaryotic. Prokaryotic flagella will have what arrangement ma? 9+0 arrangement. Eukaryotic flagella will have 9+2 microtubular arrangement. Prokaryotic flagella will have what protein? It will be having flagellin, take down parallelly, and eukaryotic flagella will have what protein? Tubulin. Fine, flagellin and tubulin. Here it is 9+0 arrangement, here it is 9+2 arrangement. In 9+2 arrangement also, the basal body of the flagella is adjoined to the cell membrane or cell wall by what? Centriole. So, it is having 9+0 arrangement only, basal body with 9+0 arrangement. Clear, right? Now, so now we are clear with two things. What are they? One is centriole arrangement, centriole microtubular arrangement, and second is cilia and flagella. The total number of microtubules in a centriole will be 9+0. 9 plus 0, and total how many ma? 27 microtubules. In cilia and flagella it is 9 plus 2 and total it is 18 microtubules. Fine, clear, right? Next. So, here you can see the structure, how flagella will be moving in this way, that is through the orientation of the microtubules which are present inside it. At the center, can you see 2 microtubules, and this is the central sheath, 9 radial spokes. To each radial spoke we have 2 microtubules, okay, 2 peripheral microtubules. They are adjoined by what? Dynein arms. Fine. Next, cytoskeleton. Cytoskeleton is a network of proteinaceous filaments which is present inside the eukaryotic cell. Okay. This gives proper shape and maintenance to the cell, and the cytoskeleton will also aid in the cell division process. Okay. Take down parallelly. Cytoskeleton is a network of proteinaceous filaments cytoskeleton is a network of proteinaceous filaments which gives cell shape, aids in cell division, and also helps in maintenance of cell shape. Maintenance of cell shape. Clear, right? Now, in cytoskeleton, the network of proteinaceous filaments, we have 3 proteinaceous filaments. Those are microtubules, microfilaments, and intermediary filaments. Okay? Microtubules be having tubulin protein, tubulin proteins. Microfilaments will be having actin and myosin filaments. Intermediary filaments will be having lamin proteins. Fine, right? Now we will look into the comparative analysis between microtubules, microfilaments, and intermediary filaments. So here if we see the comparative analysis between the microfilaments, intermediary filaments, and microtubules. Microtubules you look into. Microtubules are unbranched hollow tubules of protein tubulin. They are made up of the protein tubulin and they are unbranched, right? They have around the diameter of 25 nanometers in diameters, and they are made up of 13 protofilaments. Okay, 13 protofilaments. And at the center if you see, the core is having 15 nanometers diameter. Okay, so total microtubule diameter, it is unbranched hollow tubules. Total diameter is 25 nanometers. They are made up of 13 protofilaments, and at the core if you see, it is 15 nanometers in diameter. And this constitutes what? Spindle fibers. Microtubules are present in where? Spindle fibers, chromosome fibers, basal bodies of flagella and cilia. Okay, basal body of flagella and cilia will have 9 plus 0 arrangement only, remember that, because basal body is adjoined to the cell membrane and cell wall, that is mediated by the centriole which is present inside the cell. Functions as cytoskeleton. Okay, microtubule functions as cytoskeleton. This is required in intracellular transport. This controls shape of the cell, shape of the wall-less cells and the nucleus. This helps in the movement of nuclei during the cell division, that is during the process of catalysis, and this helps the formation of cell plate and cell movement as well. Cell plate during plant cytokinesis. Okay. So, these are the most important functions of microtubules. We have to fix it in our mind. Okay. Next is microfilaments. Microfilaments are the narrow cylindrical rods or protein filaments made up of the protein actin. Okay. These are the hollow tubules, microtubules. Microfilaments are narrow cylindrical rods or protein filaments which are made up of the protein actin. Okay. What are the functions included by microfilaments? Cytoplasmic streaming, they will give proper support, formation of myofibrils, muscle fibrils, and formation on movement of microvilli in small intestine, formation and retraction of pseudopodia in amoeba, which is the false feet, endocytosis and exocytosis. It will help in both during endocytosis, that is cell engulfing, and cell omitting during the exocytosis. Formation of spindle apparatus also, and movement of cellular components inside the cell. All these are the functions of microfilaments. And then intermediary filaments. How can you describe intermediary filaments? As they are solid unbranched filaments formed of variety of proteins. Okay, they are formed of variety of proteins. What are the major functions involved in the intermediary filaments? Formation of nuclear matrix, they support membranes, supporting array of cytoplasm, they support for contractile units, and mechanical strength, they give the mechanical strength to the nervous tissue. Clear, right? They give the mechanical strength to the nervous tissue. So, these are the set of cytoskeleton proteinaceous filaments that we have to be aware of. These are present in the eukaryotic cell only. They aid in the cell movement, cell division, and maintenance of cell shape. In specific, we have to be clear with the specific functions of microtubules, microfilaments, and intermediary filaments. Clear right now? Next. So, with this the proteinaceous structures are clear to us, both the centriole, centrosome, cilia and flagella, and cytoskeleton structures as well. These are clear to us. And from cells and tissues, what are we left with from cells, not tissues? From cell part, we are still left with what? Double membrane bound organelles. Okay, we are left with double membrane bound organelles. What are these double membrane bound organelles that we have come across so far? What are the double membrane bound organelles that we see in the eukaryotic cell? First is nucleus, second mitochondria, and third is plast. Nucleus and mitochondria are present in the animal cell, whereas mitochondria and plastids are present where? In the nucleus, mitochondria and plastids are present in the plant cell. Okay, so here if you see mitochondria and the plastids are called as semi-autonomous cell organelles. Semi-autonomous or self-replicating organelles. Semi-autonomous cell organelles or self-replicating cell organelles. First, today we will look into the structures of nucleus, mitochondria and plastids. Tomorrow we will look into the functional descriptions of nucleus, mitochondria and plastids. First, nucleus. Nucleus will be having 4 components inside it. Okay. What are the 4 components? First, if you see, nucleus is covered by a nuclear envelope. Fine. It is covered by a nuclear envelope which has pores inside it. Okay. It has pores inside it. So, it has nuclear envelope, nuclear pore, and inside which it has a nucleolus. Okay. Nucleolus and a genetic material. Fine. So, what are the 4 components which are present inside the nucleus? Nucleolus, nucleoplasm, nuclear pores, and nucleus. Fine. So, these are the 4 components that you see in the nucleus. Tomorrow we will look into the functions that are done inside the nucleus, what are the functions in the nucleoplasm and what are different shapes of the chromosomes, okay. And mitochondria and plastids. What is the structural description of mitochondria? Mitochondria is a double membrane bound organelle where it has an outer mitochondrial membrane. It can be sausage or cylindrical shaped. It can be sausage or cylindrical shaped like this. Fine. And the inner membrane of mitochondria has enfoldings called as what? Cristae. Fine. And inside the cristae you have what? Mitochondrial matrix. So, this also has 4 components. What are it? Outer is outer mitochondrial membrane and inner mitochondrial membrane is also called as cristae, and the space between outer mitochondrial membrane and inner mitochondrial membrane is called as intermembrane space, and inside which you have mitochondrial matrix. Mitochondrial matrix. Fine, right? Now, the last double membrane bound organelle is plastids. So, plastids, if you see, we have 3 different types of plastids. 3 different types of plastids include chromoplast, chloroplast and leucoplast. These are the 3 different types of plastids. Okay. So, here if you see the chloroplast structure, chloroplast is a double membrane bound organelle. Inside the chloroplast, you have membranous sac-like structures called as thylakoids. The membranous sac-like structures are called as thylakoids. Stalk of the sac-like structure is grana. Okay. Stalk of sac-like structure is grana. One grana is connected to the other grana by stroma lamellae. Okay. Is what? Thylakoid. Thylakoid is structure. So, the membrane of the thylakoid is called as thylakoid membrane or thylakoid lamellae. The stalk of this sac-like structure is what? Grana, and one grana is connected to the other by stroma lamellae. Fine. And inside the chloroplast you have stroma, right? This stroma has double stranded circular DNA. Double stranded circular DNA. This double stranded circular DNA is involved in the synthesis of world's abundant protein called as rubisco, and the double stranded circular DNA is present in the mitochondrial matrix as well, okay, which looks into the metabolic functions that are done inside the mitochondria. So, here if you see, ma, what are the double membrane bound organelles? In just today we worked out on the structural frame out of the double membrane bound organelles. Those are what? Nucleus, mitochondria and chloroplast. Nucleus has 4 components: nucleolus, nucleoplasm, nuclear pores and nuclear envelope. Tomorrow we will look into the functional aspects and the shapes of chromosomes as well. And then mitochondria, if you see, outer mitochondrial membrane, inner mitochondrial membrane also called as cristae, space in between them is called as intermembrane space, inside which you have mitochondrial matrix. And double membrane bound organelle chloroplast, inside which thylakoids are what? Membranous sac-like structures, and the membrane of the thylakoid is also called as thylakoid membrane or thylakoid lamellae. Stalk of this sac-like structure is grana. One grana is connected to the other grana by stroma lamellae, okay, and the double stranded circular DNA is present inside, both inside the mitochondrial matrix and chloroplast stroma. Here it is involved in the synthesis of world's abundant protein, that is rubisco, and here this double stranded circular DNA inside the mitochondria is involved or looks into the metabolic activities inside that organelle. Tomorrow we will finish off the cell concept by completely revising prokaryotic cell, eukaryotic cell, single membrane bound organelles, endomembrane system and double membrane bound organelles in detail, and we have to look into the microbodies as well, peroxisomes and glyoxisomes. So, that is all for today's session, and thank you.